Just a second, please. Just a second, please.
Including animals, plants, and fungi and protists. Eukaryotes are differentiated from the prokaryotes mainly by their genome enclosed in the nucleus, surrounded by the nuclear membrane, and the presence of other membrane-bound organelles. The term organelles mean little organs and include membrane-bound subcellular structures in eukaryotic cells, and they perform specific functions that are necessary for the survival of the cell. Some eukaryotic cellular organelles are mitochondria, endoplasmic reticulum, Golgi apparatus, vacuoles, lysosomes, and chloroplasts. In addition to organelles, the cytoplasm of eukaryotic cells also contains tiny particles called cytoplasmic inclusions suspended in the cytosol. The cytoplasmic inclusions are of many types present in different cells and include calcium oxalate crystals, silicon dioxide crystals, lipid droplets, storage granules of starch, glycogen, etc. The eukaryotic subcellular organelles and cytoplasmic inclusions are discussed as follows. First, let us see about the subcellular organelles. The first subcellular organelles that we are going to discuss is the Golgi apparatus. The Golgi apparatus, also called Golgi body or Golgi complex, is found universally in both plant and animal cells. They are made of 5 to 8 cup-shaped membrane-covered sacs called cisternae. In animal cells, about 10 to 20 cisternae are linked into a single complex by tubular connections between cisternae to form Golgi stacks, while in some unicellular flagellates, about 60 cisternae combine to make up the Golgi apparatus. The proteins and lipids synthesized in the smooth and rough endoplasmic reticulum bud off in tiny bubble-like vesicles that fuse with the Golgi membranes, and their contents are released internally into the Golgi apparatus where modification of proteins and lipids or fats take place before exporting outside of the cell or transporting to other locations in the cell. Carbohydrate or sugar portion of glycoproteins are commonly modified by enzymes present in the Golgi lumen by adding or subtracting individual sugar monomers. In addition, the Golgi apparatus also manufactures a variety of polysaccharides such as pectins in plant cells, and the Golgi apparatus also sort the macromolecules produced by the cell and target them for distribution to different parts of the cell. The next organelle is the plastids. Plastids are plant cell organelles and are of several different types. Leucoplasts are colorless plastids involved in the synthesis of starch, oils and proteins, while chromoplasts are yellow to red colored plastids that manufacture carotenoid pigments. Chloroplasts, on the other hand, are the green colored plastids containing the pigment chlorophyll A and chlorophyll B and are involved in photosynthesis. The chloroplast is bound by a double membrane and the space between the two layers is called the intermembrane space. Enclosed by the membrane is a semi-fluid material called the stroma that contains enzymes, DNA, 70S ribosomes and RNA. The next subcellular organelle is the endoplasmic reticulum. The endoplasmic reticulum, or ER in short, is a network of interconnected flattened sacs and branching tubules that extends throughout the cytoplasm in plant and animal cells and contains an internal space or lumen bound by a single continuous membrane connected to the double-layered nuclear envelope. Endoplasmic reticulum may be smooth endoplasmic reticulum or rough endoplasmic reticulum. The rough endoplasmic reticulum contains 80S ribosomes attached to their membrane, while smooth endoplasmic reticulum lacks them. The smooth endoplasmic reticulum plays a role in lipid biosynthesis, carbohydrate metabolism, and detoxification of toxic compounds, while protein synthesis and transport occurs on rough endoplasmic reticulum. The next organelle is mitochondria. Mitochondria are rod-shaped organelles bound by double membranes consisting of an outer membrane and an inner membrane. The space between the membranes is called the intermembrane space, and the membranes surround an internal space called matrix. The outer membrane contains many channels formed by the protein porin, while the inner membrane is highly convoluted, forming a large number of infoldings called cristae. The matrix contains the mitochondrial DNA, 70S ribosomes, and metabolic enzymes. Mitochondria are the site of cellular respiration. The next structure is cell nucleus. The nucleus is a highly specialized eukaryotic organelle that stores the hereditary material or DNA and coordinates the cellular activities including growth, intermediary metabolism, protein synthesis, and cell division. It is bound by a double-layered membrane called nuclear envelope, and the space inside the nucleus is a semi-fluid matrix called the nucleoplasm, which contains chromatin, one or more nucleoli, and other smaller components such as Casal bodies, gems or gemini of coil bodies, and interchromatin granule clusters. The next structures are the endosomes. Endosomes are membrane-bound vesicles found in animal cells formed by complex processes known as endocytosis involving the invagination or folding inward of a cell's plasma membrane to surround macromolecules or other matter diffusing through the extracellular fluid. The size of vesicles may vary, and vesicles larger than 100 nanometers in diameter are referred to as vacuoles. Endocytosis includes three primary mechanisms. Receptor-mediated endocytosis, in which cells take in molecules or ligands that bind to receptor sites extending from the cytoplasmic membrane into the extracellular fluid surrounding the cell. Then, secondly, pinocytosis, in which a cell ingests droplets of liquid from the extracellular fluid, and phagocytosis, in which a cell engulfs outside material by stretching out pseudopodia and encircling any food particles into large vesicles. Endosomes are incompletely capable of breaking down many proteins and fats, and they commonly mature into or fuse together to form lysosome to fully digest all their contents. Let us now see the next subcellular structure, that is the lysosomes. Lysosomes are spherical organelles surrounded by a single membrane, and their size and shape of the vesicles varies. The lysosomes contain the hydrolase enzymes that recycle organic materials of the cell by a process known as autophagy, in which they break down cellular waste products, fats, carbohydrates, proteins, and other macromolecules into simpler compounds and then transfer it back into the cytoplasm as simpler molecules. The next structures are the peroxisomes. Peroxisomes are roughly spherical microbodies bound by a single membrane found in the cytoplasm of almost all eukaryotic cells. Peroxisomes contain a variety of enzymes that break down toxic substances like hydrogen peroxide or other metabolites. They are a major site of oxygen utilization and are numerous in the liver of animals where toxic byproducts accumulate. Peroxisomes can self-replicate like the mitochondria. However, peroxisomes do not have their own DNA molecule like the mitochondria. The next structure are the ribosomes. Ribosomes are tiny organelles not bound by a membrane but composed of approximately 60% ribosomal RNA and 40% protein. Eukaryotic ribosomes are produced and assembled in the nucleolus present inside the nucleus. To form two ribosomal subunits, that is, one small and one large, described by their Swedberg or ACE values based upon their rate of sedimentation in a centrifuge. The ribosomes in a eukaryotic cell have a Swedberg value of 80S and comprise of 40S and 60S subunits, and 70S ribosomes consisting of a 30S and 50S subunit. In eukaryotic cells, 80S ribosomes are mainly found bound to the nuclear envelope, the rough endoplasmic reticulum, or free in the cytoplasm, while 70S ribosomes are present in the chloroplast and mitochondria. The organelles serve as the protein synthesis machinery of the cell and are more abundant in cells that are active in protein synthesis, such as pancreas and brain cells. Ribosomes carry out protein synthesis in assistance with two other kinds of RNA, that is the messenger RNA or mRNA and transfer RNA or tRNA. The next structure are the vacuoles. Vacuole is a large central water-filled vesicle enclosed by a membrane known as the tonoplast present in plant cells. The function of vacuole is to maintain the osmotic pressure and turgidity of the cell and also stores useful materials and digest waste proteins and organelles. Vacuoles in animal cells, however, are much smaller and commonly used to temporarily store materials or to transport substances. The next subcellular structure are the eukaryotic cytoskeleton filaments. Eukaryotic cells contain three kinds of cytoskeletal filaments, that is, the microfilaments, intermediate filaments, and microtubules, which maintain the structure and shape of cell and also helps in excluding some macromolecules from the cytosol. Microfilaments are around 6 nanometer in diameter, composed of two intertwined chains of the protein actin. When microfilaments are first formed in the cell, actin appears in a globular form known as G-actin, while in polymerase chain, it appears as a filamentous form or F-actin. These are more concentrated just beneath the plasma membrane and helps in cytoplasmic streaming to maintain cellular shape and in forming cytoplasmic protuberances like pseudopodia and microvilli, and also participate in forming some cell-to-cell or cell-to-matrix junctions. Microtubules, on the other hand, are hollow cylinders about 23 nanometer in diameter, comprising of 13 protofilaments made of polymers of alpha and beta tubulin. They are very dynamic, using GTP for polymerization and form the centrioles in 9 triplet sets and form cilia and flagella in 9 doublets oriented about 2 additional microtubules, commonly referred to as the 9+2 arrangement. Microtubules participate in the formation of spindle fibers during cell division and are also the major components of cilia and flagella. They also play a role in formation of cleavage furrow during cytokinesis and in muscular contraction along with myosin. Intermediate filaments are around 10 nanometer in diameter, more stable than actin filaments and are more heterogeneous in nature. Intermediate filaments are found only in some metazoan species, including vertebrates, nematodes, and mollusks. A specialized class of intermediate filaments are a fibrous network of protein called the nuclear lamins that lines the inside of the nuclear membrane. In vertebrates, most animal epithelial cells contain intermediate filaments consisting of keratins, mesenchymal and muscle cells are rich in vimentin and desmin, and neurons and glial cells contain peripherin neurofilaments and glial fibrillary acidic protein or GFAP. They function in the maintenance of cell shape by organizing the internal three-dimensional structure of the cell, anchoring organelles and serving as structural components of the nuclear lamina and sarcomeres. They also participate in some cell-to-cell and cell-to-matrix junctions. Let us now learn about cytoplasmic inclusions. Cytoplasmic inclusions in eukaryotic cells are non-living subcellular substances found in the cytoplasm. They are not bound by membranes and do not carry out any metabolic activity. Instead, they represent stored nutrients, secretory products, and pigment granules. Some examples of cytoplasmic inclusions are storage granules of starch and glycogen. For example, glycogen granules are found abundantly in muscle and liver cells. Lipid droplets or storage form of triglycerides common in adipocytes and hepatocytes. Crystalline inclusions like calcium oxalate crystals and silicon dioxide crystals present in certain cell types such as Sertoli cells and Leydig cells of the human testes and in macrophages. Pigment granules of skin, hair, retina, and specialized nerve cells in the substantia nigra of the brain. Let us now conclude the topic.
Okay, we just have understand the thing that is how the different cell components is engaged here in that different cell components are engaged in the that prokaryotic, eukaryotic, or animal cell, plant cells. So let's understand that another important area of this study is carbohydrate and lipids. So let's watch the video and understand, try to understand what is the behavior of a carbohydrate and lipids in this case.
Polysaccharides where you have mixture. It's as you can see here in this cartoon, you have three chains of carbohydrates, and you see that it is not one single monosaccharide that continuously repeats. For example, in starch, you have glucose continuously repeating glucose after glucose. So instead, here you have a mixture of two different kinds of sugars, and that's why they are called heteropolysaccharides. So here you have N-acetyl glucosamine and N-acetyl muramic acid. So N-acetyl muramic acid, yesterday we saw, these are the modified monosaccharides. So this has a lactic acid group attached to carbon 3, while the carbon 2 has the amino group that is acetylated, N-acetyl muramic acid, and then you have N-acetyl glucosamine. So these two monomers alternate in these long chains, and multiples of these chains are held together by these 4 amino acids attached to one of the carbons. So alanine, glutamic acid, lysine, and alanine. And these chains, these 4 peptides are held together in gram-positive bacteria by this pentaglycine cross-link. So this sort of a structure forms the cell wall of bacteria. Many bacteria, like both gram-negative and gram-positive, have these sort of structures called peptidoglycans. So here, so the long chain of carbohydrates are called glycan. Okay, so gly is the generic term for any carbohydrate. So if it is only glucose, you will say glucan. So gly is a generic thing for any sugar. So glycan means a long chain of monosaccharides. So since they have peptides attached to them, they are called peptidoglycans. So these peptidoglycans form the major cell wall component of bacteria. So in gram-negative, instead of pentaglycine, these amino acids are directly joined. Like this alanine is joined either to the lysine or to alanine, and so on. So this is one of the important heteropolysaccharide, and it is important in studying many bacterial infections as well, because some of the drugs are focused on destroying these peptidoglycans. For example, lysozyme, which is abundant in the tear drops, the tear secreted in our eyes, which protect the eyeball, the outer part, cornea, lens, etc., is protected by the liquid, your tear, and the tear is rich in an enzyme called lysozyme. And this lysozyme cleaves this glycosidic bond, this beta-1,4 glycosidic bond of the peptidoglycan. So as a result, the bacterial cell wall gets broken and the bacteria dies. Okay, so this is how your tear affects your eyes from a bacterial infection. So the next set of heteropolysaccharides we are going to see are called glycaminoglycan. So glycosamine. So we already know glucosamine. You know, this amino group attached to usually the second carbon. And that I told you yesterday is usually acetylated. So N-acetylglucosamine. So we saw in chitin you have the N-acetylglucosamine having beta-1,4 linkage. So this sort of a structure, generic, when it is glucosamine, you call glucosamine. If you are referring to any other hexose or any other monosaccharide, you simply got glycosamine. The focus is on this amino group. And when you have long chains of them, they are called glycosaminoglycans. Okay. So here we are looking at the four major ones, and they all have very important functions, and that is why we are interested in learning the structures here. The first one we are learning is called the hyaluronic acid. When it is in the dissociated, called oxidase to carboxylic acid, you call uranic acid. If the anomeric carbon, the aldehyde group is oxidized to carboxyl group, we call that as aldonic acid. So yesterday I told you that. So this is a uranic acid. So hyaluronate already suggests that it has uranic acid group. And this one, you have attached by glycosidic bond to N-acetylglucosamine. So this glucuronate N-acetyl glucosamine keeps repeating like 50,000 of them. Okay, so the disaccharide is the repeat unit, because if you take this alone, it doesn't describe the whole polymer constituents of the polymer. But if you take both of them together, then you have them repeating. So it is a disaccharide repeating. Disaccharide is the repeating unit of this polymer. And it is very, very high molecular weight chain, because it can have as many as 50,000 monosaccharides in them. So given all the hydroxyl groups and carboxyl group, this is highly polar and provides multiple points for interactions. Okay. And due to long chain nature, as well as due to this charged and polar groups, they form good lubricants in our synovial fluid of the joints. If you take your knee joint, that's a good example where you find plenty of synovial fluid. So since the time you are born until you die, nobody is going to open up and keep lubricating. Okay. So it's permanently lubricated. The liquid produced by the cells around there, they secrete this hyaluronic acid, and that gives a good lubrication. It's kind of a jelly, thick liquid, and that consistency comes primarily from this long chain and with multiple polar groups in them. And similarly, the vitreous humor of eye. So behind your lens, the eyeball is filled with a jelly-like consistent liquid, and that is primarily composed of hyaluronic acid. Okay, so that gives a protective and cushioning effect to the eye. So this sort of a role is very important, and this is also present in cartilage as well, where it provides tensile strength. So that is the function of hyaluronic acid. And another such glycosaminoglycan are the chondroitin sulfate. So these are much shorter than hyaluronic acid, as you can see here, 20 to 60. And these are sulfated, so you have sulfur group attached. And instead of N-acetylglucosamine, this is N-acetylgalactosamine having a sulfur group attached here. And these are usually not present in isolation as carbohydrate. Instead, it is usually attached to proteins, forming what are called proteoglycans. So we will learn about it in a short while. In a couple of slides, we'll get to proteoglycans and learn about their importance. And These molecules, where the core protein is a smaller component of the whole molecule, the major component are the carbohydrate part, which is multiple chains of chondroitin sulfate. So these are present in cartilage. For example, if you take your ear lobe, the outer part of your ear, that has thick consistency. It is not like your skin, okay? And the same time, it's not like bone. And that sort of consistency is provided by these proteoglycans containing chondroitin sulfate. Tendons, the ones that joints, muscles and bones, ligaments, again, similar structures. And the walls of aorta, the main artery supplying to heart. So they all get their tensile strength primarily from the chondroitin sulfate attached to proteoglycans. And you look at the charge available here, carboxylic acid, sulfuric acid moiety here. Then you have plenty of polar groups, multiple hydroxyl groups. So all of this provide multiple points for various covalent, non-covalent interactions with other molecules in these structures. Okay, and that sort of multiple interactions enabled by this structure is what provides the tensile strength for cartilage. Okay, so that is chondroitin sulfate. A similar molecule is keratin sulfate. When you think of horns, hooves, hair, nails, claws, and our own cornea, they all have this keratin sulfate. So there again you have a similar structure. Here the uranic acid component is not there. Okay, so these two have uranic acid and this doesn't have. And instead it has usually galactose attached to N-acetyl glucosamine, where the sixth carbon has a sulfuric acid attached. Okay, so it's an ester between an alcohol hydroxyl group and then the mineral acid, sulfuric acid. So you have strong negative charge. So these strong negative charges repel, and as a result, these molecules remain in an elongated, extended structure instead of being coiled. Since it's an introductory class, I didn't get into the multiple conformations possible for cellulose, starch, etc. So you don't need to worry about it. But here, because of the repelling charges, we just briefly visit its structure. So here the long fiber remains in an extended form due to the charge repellent. So the primary aspect of this structure is that these multiple groups that can form covalent bond or electrostatic interaction or other hydrogen bond kind of non-covalent interactions, they provide multiple contact points for various molecules to interact. And finally, the ultimate result of such interaction is the tensile strength of these tissues. So you look at hair, or you take a horn, or take your nails. That is probably the easiest. So the nails have a certain consistency, and that is because of keratin sulfate, and they also contain other fibrous proteins that is also responsible for this. Then the fourth one we are going to learn about are the heparin. So heparin are present in our blood, and that prevents blood clotting. So when you collect blood sample from patients for any analysis, usually the tube in which you collect contains a little bit of heparin, so that the blood doesn't get coagulated, doesn't become clot, doesn't become solid. Okay. So normally when you have an injury, when there is a cut and the exposure of the blood to the external environment, that exposure, like contact with the non-endogenous structure, triggers a process that results in blood becoming solid-like structure. So that seals the wound. Okay, the blood clotting is an essential phenomena in controlling bleeding when you have a cut injury. Okay. So when you don't want to have that, like for example, the circulating blood shouldn't become clotted, and that is usually prevented by the presence of heparin. And heparin itself is a polysaccharide, so 15 to 90 repeating units. It has a lot of sulfur groups attached to it. So that's the primary structural component of heparin, structural feature of heparin. Okay. So iodose is one of the isomers, epimers of glucose. So here you have either glucosamine, the sulfated glucose, or glucuronic acid. GLCA is glucuronic acid with 2 sulfate groups, or you have the carbon 2 sulfur. That's what this name indicates. Or you might have an iodose having uronic acid like carboxyl group at the 6th carbon and the 2nd carbon having a sulfur. So this is an important feature, primarily the sulfur attached, the sulfate groups being dominant feature of this molecule. So these are the primary glycosaminoglycans. There are other minor ones as well. So primarily these are long chain polysaccharides having charged acidic groups on them, and they either function as lubricants or provide tensile strength to structures like cartilage, tendon, and bone, horns, nails, and so on. And these molecules are actually very important when you think of making extracellular matrix artificially for tissue engineering purposes. Okay, so the people who study tissue engineering, they learn a lot more details about these glycosaminoglycans and other extracellular matrix proteins. Extracellular matrix is the cementing material among the multiple cells. Okay, what is there in the space among the cells? When you have a tissue with multiple cells, what is there between 2 cells? And that part is what you call as extracellular matrix. That acts more like, in the simplest, crudest way, is like the mortar that you use to stack up bricks in a wall. So you're not simply stacking up the bricks to build a wall, and if you push, the bricks will fall. But you use mortar to seal the bricks. So the bricks are held tight. They are stuck to each other. And that sort of a function among cells, if you think individual cell as bricks, then extracellular matrix is like that mortar. So not only that, extracellular matrix is very important for cell-cell signaling as well, and in making sure the cells migrate in the right direction to take the right shape to form the different organs. So extracellular matrix is a lot more than the mortar that you encounter in the walls. And to a great extent, that property of extracellular matrix comes from these glycosaminoglycans. So here, as we move on, you are slowly seeing how we started with a few elements, seemingly simple molecules, and stacking them, joining them in multiple ways, how we are able to build functional complexity in the living system. So this is the beauty I want you to appreciate and focus attention while you learn these structures. Okay. Do not think, oh, I have to memorize this structure, which has the sulfate, which has the carboxylic acid, what is epimer. Don't get distracted by that exam-oriented thought. Instead, look at the complexity coming from the simplicity in living systems. So the next molecule you are going to learn are the proteoglycans. Okay, so we are going to learn later what are called the glycoproteins. So glycoproteins, where the protein is the major thing, the glyco, that is carbohydrate part, is a minor thing. By contrast, proteoglycans, glycan is the main thing. It's long polysaccharide, and the protein part is the minor thing. So let us look at its structure, then we'll get a good understanding. So I already told you proteoglycans contain chondroitin sulfate. So usually the chondroitin sulfate polysaccharide is attached via a trisaccharide linker, galactose, xylose, to a serine hydroxyl group on a protein called the core protein. Okay, so proteoglycans have a protein component which we call as the core protein, and to one of its serine residues or multiples of its serine residues, usually in this context, that serine has a glycine and any amino acid represented as X and another glycine. Towards the carboxy terminus, such a serine is potential for attaching this chondroitin sulfate via this trisaccharide linker. Okay, so this is the structure of a proteoglycan. And again, as I told you earlier, in many cases, the biological activity is the provision of multiple binding sites. See, this is uranic acid, a carboxyl group, and then it has hydroxyl groups on the glucose moiety itself. And similarly, here you have got an amino group and acetyl group, therefore another carbonyl group, and fourth carbon has a sulfate, and so on. So like this, you have got multiple binding sites, rich in opportunities for hydrogen bonding and electrostatic interactions with other proteins of the cell surface or the extracellular matrix. Okay. So proteoglycans are major components of the connective tissue, such as cartilage. I told you a good example, cartilage, your outer earlobe, the nose as well. So you touch and feel your nose, how it feels. In which there are many non-covalent interactions with other proteoglycans, proteins. There are proteins present in extracellular matrix, the main one being collagen, and then fibronectin. So these proteins are all interconnected among themselves and to these carbohydrate molecules, which are often anchored into the membrane itself. Like for example, this core protein may be an integral membrane protein, where hydrophobic part of the chain interacts with the lipid bilayer, and therefore it is actually anchored in the membrane. So that is how the cells are cemented with the extracellular matrix. So they provide strength and resilience to the overall structure. So here is an example of proteoglycan, which is an integral membrane protein. So here the core protein contains hydrophobic side chains in a short sequence. Usually about 10 to 12 amino acids, hydrophobic amino acids are enough to get anchored in the membrane, where the hydrophobic side chains here interact with the aliphatic chains that remember, we already know phospholipid structure. The glycerol part has the phosphate and therefore it is hydrophilic, and the other two hydroxyl groups of glycerol have esterified with the long chain fatty acids. These are the yellow tails here. So these are hydrophobic. So these hydrophobic tails of the phospholipids interact with hydrophobic amino acids on this short chain, and that is how this protein is anchored in the membrane. And such proteins which are integrated into the membrane are called integral membrane proteins. And one end of it, the amino terminus here, is extracellular. And in the extracellular region, it has multiple tails of these molecules. Like in this particular example, this molecule has 2 chondroitin sulfate shown in yellow and 3 heparan sulfate. So, and these now interact with collagen, fibronectin, and so on, and they form a tight mesh network from intracellular to the extracellular environment. So if we take one chain of this chondroitin sulfate and look closely, what you have is a region where you have the carbohydrate moiety strongly sulfated. And then you have this gray area where you do not have sulfation. Instead, they are simply N-acetyl groups, and therefore they are called Na domain. So S domain because sulfate, and then N-acetyl, so Na domain. So they alternate in this long chain. So this is an example of a proteoglycan structure. So the protein itself is called the core protein. So there are certain situations where the extracellular matrix contains really a humongous molecule where you already learned hyaluronic acids can have as many as 50,000 repeating disaccharides, meaning 100,000 monosaccharides. Okay, a long chain. So these are uranic acid. Remember, we have seen that structure just a couple of slides ago. And to them, you may have core proteins attached. And these core proteins, unlike the earlier one we saw, these are smaller molecules, okay, not very big molecule. Whereas this particular core protein is very large and it's called agrican, written here, agrican. See, these are long rod-shaped molecules, and instead of having 2 chondroitin sulfate and 3 heparin sulfate, these have several of them attached. Okay, and such long core agrican with multiple chondroitin sulfate and keratin sulfate attached are again connected to this long hyaluronic acid and held in place by link proteins. And such long structures are a very, very integral component of extracellular matrix, providing the structural characteristics of the extracellular matrix. So make sure you remember this structure. So this is how the bird's eye view big structure looks. But if you look at each one of them, they are core proteins. So you have multiple proteins here in one single molecule. And each protein has multiple this shorter heteropolysaccharides. And all of them attached to one long hyaluronic acid. So this gives you an overall view of the extracellular matrix environment. So this is your lipid bilayer. Okay. So this is the inner leaflet and that's the outer leaflet. So the surface of both of them are hydrophilic because of the phospholipids head group. And they have, this is inner side of the cell where you have the cytoskeleton made up of actin and tubulin, which we haven't yet learned. You will learn it in cell biology later. So these provide structural stability and integrity to the internal environment of the cell. They are directly connected via these protein molecules called integrins. Okay, the integrins are integral membrane proteins that are the main components for connecting the intracellular structural molecules like this actin filaments, which is entirely made up of protein. So here you do not have carbohydrate. So directly it connects them to the extracellular structures like fibronectin and to proteoglycans and collagen. And these are all cross-linked because you have multiple functional groups available for such interactions. So all these multiple interactions of really large molecules make a mesh-like structures, and that is what is our extracellular environment is. And that is very critical for the function of the cell. And it is very important for cell migration during organism development. And they are also important for cell-cell signaling. So they have multiple roles. Just do not view this as if it is analogous to the cement or mortar used for holding bricks in the wall. Okay, so that is about our discussion on glycosaminoglycans. Next, we are going to a set of molecules where the carbohydrate part is a minor component, nevertheless functionally very, very important. And the major thing being either protein in case of glycoproteins or glycolipids, where it is going to be lipids having carbohydrates attached. First, let us look at the glycoproteins. So here the glyco part is actually oligosaccharide. It's not long polysaccharide. And usually there are two types of linkages between carbohydrate and proteins. They are either via the serine or threonine hydroxyl group. Okay, so this is the side chain. So this is the peptide, polypeptide in which you have the serine. So amino group, carboxyl group. So this is in peptide bond with another amino acid. Here the carboxyl group is in peptide bond with the other one. So this is the alpha carbon. So you have the side chain CH2OH, and that is in linkage with anomeric carbons hydroxyl group of N-acetyl galactosamine in this particular example. So this sort of a linkage we call as O-linked, because the atom that connects the carbohydrate to the protein is oxygen. On the other hand, N-linked ones have the amide group of the asparagine side chain. Asparagine, here you have CH2, CONH2, and that is in linkage with the anomeric carbon. Here you have N-acetyl glucosamine. So it need not be this particular carbohydrate moiety alone. It could be any, but the point is this is an N linkage via asparagine. Here it is O linkage via serine or threonine. So these are the kinds of linkages that we see in glycoproteins. And here are some examples. So these are specific carbohydrates attached. Each one is color coded and explained here. Mannose, galactose, N-acetyl neuraminic acid, fucose, and so on. So you see here they all have specific sequences, and these specific sequences can therefore have specific codes. Each one can be like a pin code, and therefore they are recognizing structures. So if a cell surface protein has a certain carbohydrate chain attached, that could be a pin code or an identity for that particular cell. So therefore these oligosaccharides of glycoproteins function, or confer important function in cell recognition. In terms of molecular recognition, they function as address tags. Okay, and that is how they are important in our blood group as well. But in blood group, it is not glycoprotein; it is glycolipid. I will come to that in a minute. And then now we are looking at the lipid attachment. So here the structure looks complex, but it is already very familiar to us. So it is just that the Haworth formula, the ring is shown in a this sort of a shape, because this is the way it exists. But otherwise, each corner is the same. This sort of a structure. Okay. So we call that as chair and boat conformation. So this is a chair conformation. So boat conformation will be this also bent upward. That would be boat conformation. So you don't need to worry about the conformation for an introductory course. So here you have this N-acetyl group. And here instead of uranic acid, a carboxyl group or a sulfate, here you have phosphoric acid attached. That is all. Otherwise it is just regular N-acetyl glucosamine, and this is another N-acetyl glucosamine, and they have multiple fatty acid moieties attached. So if you count, there are 6 of them attached to 2 N-acetyl glucosamines, and one of them have a long chain of polysaccharides. And this is where this name polysaccharide comes, and this is what lipopolysaccharide. So these are components just like the peptidoglycans that we saw these are components of bacterial cell walls. So these are primarily present in gram-negative bacteria like E. coli, which is commonly used in the lab. And certain strains of E. coli are pathogenic as well. So they have this in their cell wall. So lipopolysaccharide. This is like space filling model for the same, in which the sixth one is not visible in this orientation. So this is the structure of lipopolysaccharides. So now I told you that these carbohydrate moieties, which are in small number oligosaccharides, attached to proteins as well as lipids, can provide address tags, and there could be other proteins that could bind to the specific sequence of carbohydrates, and therefore they could be the recognizing molecules. And such proteins that recognize specific sequence of oligosaccharides on the cell surface glycoproteins or lipids are called the lectins. Okay, so lectins are proteins that read the sugar code. So the sugar code being that specific sequence. You know, do you have this sequence, or this kind of a sequence, and so on. So it can be really, there is no limit to it. You can generate a variety of address tags. And so you could have specific proteins, each recognizing a certain sequence of sugar, and that's why we call it as they read sugar code, and they mediate cell and many molecular recognition in biological processes. So shown here is an example where this is a blood vessel in which blood is circulating, and in the circulating blood you have these immune cells called the T lymphocytes going through. And when you have an injury on an infection that leads to inflammation, and these cells go there to take care of that. And how do they identify the place and go there? That is because as they keep going through the bloodstream, their surface molecules, this carbohydrate extension of the glycoproteins, are bound by molecules, the lectins called P-selectin in this case. And when they bind to P-selectin, then their flow is slowed down. And then when they detach and move, they slowly roll along the walls, and that's why it's called rolling motion. At the site of inflammation, you have many more of these interactions, okay, many more integrins and beta-selectins binding to different cell surface glycoproteins, and they stop. And once they stop, then they migrate through the endothelium and the blood vessel wall into the tissue where you have inflammation to take care of the infection there. Okay, so that is how this recognition property of oligosaccharides of glycoprotein and the proteins that bind to them, like in this case beta-selectin, are important in mediating biological process. Many processes. In this particular example, the process is immune response to an infection or a site of injury. So that gives us a really brief introduction into carbohydrates. Probably in a couple of classes we have finished. Otherwise, if you want to learn carbohydrates in detail, we can actually have a course on carbohydrate. And that sort of study of carbohydrates we call as glycobiology. There are textbooks on glycobiology, there are journals on glycobiology. So there is a lot of things to learn about carbohydrates. But since it's an introductory biochemistry class, we end our discussion on carbohydrates there. At least we know what is what in carbohydrates, what's the structure like, what's the function. So we know if we want to learn further what we need to do. So that's all the aim of this course. Next, we will get a brief similar introduction to lipids. Okay. So already I am warning you, it is lipids 1. So there is going to be lipids 2 as well. So here are names, but they look a lot. But when you go one by one, they're all very logical and easy to remember structures. So first, let us take fatty acids, which we are very familiar with. I'm not even going to have a big cartoon of their structures because you already know them. So they are long chain aliphatic acids. So you have a carboxyl group, and then you have a long chain of carbon where the carbon valency is satisfied with the hydrogen. So you have CH2, N number, and then finally a methyl group. So that is what is fatty acids. So these are hydrocarbon chains with carboxyl group at one end. And here, for example, I'll get into nomenclature right here because it's already there. 12 means you have 12 carbons. So the carboxyl carbon is number 1, then you continue to number towards the methyl group. And this colon and 0 means the degree of unsaturation. How many double bonds do you have in this chain? So in this particular molecule, it is 0. Okay. On the other hand, if you look at it here, you have 1 double bond, 2 double bond, 3 double bond, and 4 double bonds, and so on. So that is one thing. And second, where is the double bond is indicated in the brackets with this delta sign and a superscript of the carbon number where the bond is. So for example, delta 9 means delta 9th carbon is in double bond linkage with the 10th carbon. So here you have 1, then 7, so 8, then the 9th carbon. So 9th carbon has a double bond with the 10th carbon. That's what it means. And when you have 2 of them, then you say 9, 12. All right. So when you have multiple double bonds, you indicate those numbers as shown here. So the names again, the IUPAC PAC names are extremely simple names. So decanoic meaning 10. Dodecanoic means 2 more double, that is where DO comes. So dodecanoic means 12. Tetradecanoic means 14. Hexadecanoic means 16, and so on. So I'm not going to expect you to remember these names or the logics of it. All that you need to know is these are long chain fatty acids. The most common ones are like 16 carbon, 18, and 20. These are the most common ones. Like for example, palmitic acid, which is abundant in palm oil, and that's where the name comes. So that is hexadecanoic acid. Stearic acid, which comes from hard fat like the beef. Beef has fat which is like more fatty than the butter, and so that is 18 carbon stearic acid. And arachidonic acid comes from this plant legume arachis. That's where that name comes. And so that is 20 carbon. So these 3 are the main ones. Particularly the unsaturated ones our body cannot make, and they are important via the blood. And their melting points depend on, they are usually solid at room temperature. Their melting point depends on the chain length and the degree of unsaturation, how many double bonds, etc. So they are usually not soluble in water because they are very hydrophobic. So they are soluble only in hydrophobic solvents like organic solvents. Okay, so this gives you space filling model, perspective diagram. And when you have a double bond, the rotation between these 2 carbon is restricted. As a result, you have a rigid orientation of the 2 sides of the chain, and leading to a fixed rigid structure like this. So as a result, these molecules, unlike the fully saturated ones, as you can see here, they can compactly pack. These don't pack very well. So as a result, these molecules tend to be liquid at room temperature while these are solid. So butter, for example, rich in saturated fat is solid at room temperature. Beef fat, which has not only saturated fatty acid, it's longer chain as well. That is really solid, hard solid at room temperature. Like butter is soft while beef fat is not. So only when you melt it becomes a liquid. On the other hand, many of the plant oils, vegetable oils, palm oil, groundnut oil, gingerly oil, etc., they have a lot of unsaturated fat with these sort of structures, and due to that they are liquid at room temperature. Okay, so that is one important point to consider. Then usually these free fatty acids, they travel around in our blood bound to proteins because these themselves are not soluble in water, so they move around primarily associated with proteins. And usually they are not found in the free form. Instead, they are esterified with the glycerol, with the 3 alcohol groups of glycerol, so they form triacyl glycerol as shown here. So this is the glycerol molecule with 3 fatty acids. So one of them in this particular cartoon has 2 double bonds. So we call this structure as kinks, Kinks, okay, kinks. This becomes very important when we go to lipid bilayer. Okay, so I'll talk about lipid bilayer in a minute. But let us complete this triacyl glycerol part. So this is the form in which the free fatty acids are usually present. So as you can see here, there are no oxygens or any polar thing. So these are completely reduced, and therefore when you oxidize them, there is potential for a lot of oxidation and therefore a lot of energy liberation. So these are very compact storage of energy. Compare it with glycogen or starch, where the carbon atom of these monosaccharides have hydroxyl groups. There are a lot of, they are basically polyalcohols. So their ability to, they have limiting potential for oxidation. They are not fully reduced. So as a result, the amount of energy available per unit weight of carbohydrate is about approximately half of the energy available from this. So that is why your body tends to convert the extra calorie that you eat into triacyl glycerols and stores in adipose tissue. A common place is the adipose tissue around your belly, and that's why one gets a pot belly like structures, or subcutaneous all over the body under the skin. So that is triacyl glycerol. It is important storage molecule, energy reserve. And here is an example of 4 major adipocytes. These adipocytes are the cells which are fat filled.
That's fine. This is about the carbohydrate and lipids, that lipid and carbohydrates. Now the another important fact in this particular that unit is protein and nucleic acids. Let's understand protein and nucleic acid.
The amino acids are the building blocks of peptides and proteins, whereas the nucleic acids are the ones which contain information about the proteins. The deoxyribonucleic acid, that's DNA, contains the blueprint of the protein in terms of the genetic code, whereas another nucleic acid, that is RNA, is the one which participates in translating that information from the DNA to the formation of protein in the cell. So in this whole thing, the proteins are the core of it. That's a central theme over here. And just taking another perspective, we know proteins are diet, and because that is required in our diet, because that is very essential for the normal functioning of the body, because proteins play enormous amount of functions in the body. There are a lot of biological functions which they perform. Now this whole series will be built up as follows. That is, to begin with, we will start our discussion on amino acids, and we'll focus, we'll spend a lot of time on understanding different dimensions, different aspects of amino acids. What are they, how do we represent them, and then how do we synthesize them, and then many more things about them. Thereafter we will see that how do we move from amino acids to peptides, and then to proteins. We will talk about the structural and functional part of it to some extent there, and then towards the end we will give a brief account of the structural aspect of the nucleic acids. So that is how we are going to talk about in this whole series there. As regards today's session goes, today's session is going to be focusing primarily on amino acids. We will start about the session by talking about amino acids in terms of what are they, what is their structure, and then how do we represent them. And also we will talk about the nomenclature, the traditional nomenclature of alpha amino acids, that means on the what basis have the names been given to them. Then as regards the representation of alpha amino acids go, we talk about different kind of symbols given to them, the 3-letter symbols and 1-letter symbol, and so on and so forth. And then we will spend a lot of time on classification of alpha amino acids. We will talk about different bases of classifying amino acids, and then see how do we classify them, and then what is the significance of such a classification. And towards the end we will try to sum up what we do in today's session. Okay, now let's see what are amino acids. We have just seen, we just mentioned earlier that amino acids basically are the building blocks of the proteins, but that is one bit of it. But if you really ask me, the number of amino acids known run into hundreds, and most of these happen to be alpha amino acids. These occur either as free or in the combined form, because we know that the amino acids exist as peptides, proteins, coenzymes, hormones, etc. So in the wide variety of combined forms, they are available in the cell. Further, out of this whole lot of hundreds of amino acids, there are 20 of these are the ones which are fundamental to the sustenance of the life forms, because they are the ones which are the core of the amino acids which we really want to focus attention on to. So in this whole series of lectures, we will primarily focus on to these 20 alpha amino acids. We will talk more about it as we proceed. Now if you look at, just for sake of it, the general structure of alpha amino acid, because we know that alpha amino acid, as the name suggests, is an amino acid, that means a bifunctional molecule. The bifunctional molecule is such that is, this amino group happens to be attached to a carbon atom which is alpha to the carboxylic group. So that is how it looks like. That means we have a carboxylic acid. So the carbon atom next to this, say we look at it, this is my functional group here. So typical IUPAC nomenclature. So we have a functional group, the carbon atom next to that is alpha, the still next is beta, still next is gamma. So in case of alpha amino acids, what we have is, we have an amino substituent at the carbon atom which is alpha to the carboxylic group. That is why the name alpha amino acids. Further, because if you look at this carbon atom here, this is attached to 4 different groups there. So this is carboxylic acid group, this is NH2, this is just a simple hydrogen atom, and this is R. R basically represents what is called as a side chain, because it is R which differentiates all these 20 different amino acids, because for all of them, this part of it will remain the same, whereas this R will keep changing. By changing R, we go from one amino acid to another one. So basically all amino acids are differentiated on the basis of the side chain in them. We'll talk more about it as we proceed a little further. Okay. Now, as regards to nomenclature, how do we name them? Typically, traditionally speaking, the names of amino acids have been given by their discoverers, and primarily they base the names on their sources. For example, the glycine. Glycine has been so named on the basis of the fact that it happens to be sweet in taste. In Greek, glykos means sweet. So that's how the name was derived. Its source, casein. Casein basically is the milk protein, which is what is found in cheese. So tyros again in Greek refers to cheese. So since tyrosine was found in casein, which happens to be the major protein in cheese, so that's how the name was given to be tyrosine. Further, tryptophan, it basically is the one which derives its name from its source again, that is trypsin, which is an enzyme, and in Greek, phenin means to appear. That means this particular amino acid appears in the enzyme called as trypsin. Similarly, we have got lysine, which derives its name from a tendency to participate in the catalysis part of it, primarily the lysis. That is, in German, lysine means breaking down, lysis, and so on and so forth. So that's how the traditional names have been given. But nowadays, even IUPAC accepts these traditional names because that's how do we go about it. But also for all these amino acids, we have got formal names in terms of IUPAC nomenclature. Okay. Now, so there are 20 amino acids, as I mentioned earlier. They are the ones which are the most crucial ones. We'll talk more about it. So it's just for sake of it, I given a table here which gives you the names of all these 20 amino acids. We don't have to bother much about it because we'll coming across these very, very often there. Okay. Now, out of these 20 amino acids, cysteine is the one which is unique. I just want to make a brief mention here about it because this has a role to play in the structure of proteins. And the uniqueness here is basically this is the only one amino acid in which the side chain contains a sulfahydryl group, that is SH group. And this SH group is such that it gives a very unique property to this amino acid cysteine, that is, this can undergo a reaction of this kind, whereas in the presence of mild oxidizing agent, so two molecules of cysteine can undergo a covalent linkage with these two SH groups here, SH group here, SH group here. They undergo a covalent linkage and give you a very, very significant disulfide bond. So this is a very pertinent feature of this particular amino acid only. So as I just mentioned, that this disulfide bond formation has a very significant role in imparting the structure to the protein, because there are a lot of proteins which rely heavily on the formation of the disulfide bonds on their overall shape and structure. We'll talk more about it maybe in future lectures there, where we talk in details about the protein shapes. Okay, let's move a little further, and then besides these 20 amino acids, there's some of them which again are found in the cell. They take part in a lot of biochemical processes, but and they're very, very common also, but they are not the ones which are genetically coded. We'll talk about it a little while from now. But this some other alpha amino acids, they are the ones, say ornithine and citrulline. They are very, very crucial in terms of metabolism. Maybe at some stage in biochemistry we learn about metabolic pathways. We find that this ornithine and citrulline are very, very crucial molecules there, which basically are kind of amino acids. So ornithine is something which is similar to lysine. It has got one CH2 group shorter in the side chain here, whereas citrulline actually happens to be a derivative of ornithine. Moving further, another example is say thyroxine and triiodothyronine. These two are very, very important hormones which are found in the thyroid gland, and they play very, very important role in the body there. Yet another example is that of 5-hydroxylysine, which actually is found in collagen. Collagen is again a very important protein, which is a structural protein. So that is the kind of structure of this. This basically is a small derivative, that means at 5 position of lysine, 5 carbon here. So we have a hydroxyl group there. So it's 5-hydroxy lysine. So there's some, just for sake of it, I mentioned a few amino acids which are very common, but they are not the ones, the core of 20. Okay. Now, how do we represent them? How do we represent alpha amino acids? There are two ways of doing it, because typically the amino acids have got names which are very large. If you want to talk about peptides or proteins, if you have to write the names or the sequence of proteins and all, it becomes very tiring or cumbersome to write them. So what we do is, there's some kind of a notations have been given to them, and there are two kinds of notations. The first one happens to be what's called as 3-letter symbols, and the second one is 1-letter symbols. They're the ones we'll talk in little details about both of them. Let's start with the 3-letter symbols. The 3-letter symbols, basically it is derived from the trivial name of the amino acid, and what we do is we pick up the first 3 letters of the same. That means we take the common name and take the first 3 letters, and they taken in such a fashion that is, the first letter is taken as capital and the other two are taken to be of lowercase letters. For example, if I want to talk about glutamic acid, now glutamic acid will be represented as GLU, G capital, L and U small. If you write GLU like this, all caps, it is incorrect. If you write all smalls, it again is incorrect. So 3-letter symbol basically is the first 3 letters of the common name, and first letter of the 3 will be the capital one. Now, when you write, say if I write GLU, basically I am writing glutamic acid, I am representing glutamic acid. When I am representing glutamic acid, I am representing it in terms of a unsubstituted amino acid, and they basically, as I mentioned earlier, are used to save space while representing peptides and all. Okay. Now, let us look at the 3-letter symbols here. What you find is, most of it is very, very obvious. Say alanine. So, first 3 letters are A, L, A. So, we have symbol A capital, L small, A small. Similarly, arginine, aspartic acid, and so on and so forth, because most of them are very, very straightforward. Out of 20, 16 are very straightforward. But the question comes up is, when you talk about, say, asparagine. Now, asparagine cannot be given a symbol as ASP, because ASP has been given to aspartic acid. So, aspartic acid and asparagine, both of them have got the first 3 letters to be common. So, in this case, what has been done is, it has been given a special symbol as ASN. N has been picked up from here. Similarly, there is a clash between glutamic acid and glutamine. So, glutamic acid has been given GLU. So, glutamine cannot be given GLU. So, it has been given GLN. So, that is how these two are different here. Thirdly, another clash comes up is when we talk about isoleucine. In case of isoleucine, iso could not be given to this as a name here, because iso actually is a prefix, which is a very common prefix. So, it was found to be leading to some kind of ambiguities at some stage. So, it has been given a special symbol, start with I, which is given as capital, and then the first 2 letters of leucine part of it has been taken as this. So, ILE is the symbol for this, and rest of them are straightforward. And then this comes tryptophan. Now, for tyrosine, the symbol happens to be TYR, which is very obvious for the first 3 letters, but for tryptophan, the symbol could have been TRY. So, there was very likelihood of confusion between TYR and TRY. So, it has been thought apt to give it a special symbol as TRP. So, tryptophan. So, it sounds that way, so you do not make mistake between tyrosine and tryptophan. So, there are 4 symbols which are, they are not the first 3 letters as defined it to be. So, that is how we have defined the 3-letter symbols. So, that is how do we represent these amino acids, because we do not write the full names always. Now, as regards 1-letter symbols, so this again 1-letter symbol, because what happens is, if I use 3-letter symbols, even that also becomes quite cumbersome, because if you look at some peptides and proteins, it may run into, say, a few hundreds of amino acids. So, if I write the names of 100 of them, so even 3 letters are going to consume a lot of space and time. So, what has been thought of is, we give 1-letter symbols to them. So, in 1-letter symbol, each alpha amino acid has been given 1-letter symbol, and that, as the name suggests. So, this letter symbol happens to be the first letter of the common name. So here again that same issue will come, that there will be more than one amino acid starting with the same alphabet. So from this point of view, so 11 amino acids, the symbol happens to be the first letter and it happens to be the capital letter there. But for the rest 9 of them, distinct symbol have been assigned so that there is no ambiguity of any kind. Let's look at the symbols, but before that let me make a statement here. These 3 letter symbols are quite straightforward. We know that the first 3 letters, barring a few exceptions there, but 1 letter symbols are somewhat less obvious because for 9 of them we have to give special symbols there, and they are not the ones which are coming from the roots of the name here. Let's look at the symbols. So they are the symbols for this. So we find that for 11 of them, that these blue ones here are very straightforward, but the red ones here are the ones which have been given specially for them. Say for arginine, so it could not have been given A because A has been given to alanine. So arginine, it has been given a symbol as R because as it sounds arginine, so that's how it's been given symbol as R. That's one way of looking at it. Say for aspartic acid, again because there are 4 of them starting with A, so we could not give A to 4 of them, so these 3 have been given special symbols there. Aspartic acid has been given D, asparagine has been given N, and so on and so forth. Glutamic acid has been given E, glutamine has been given Q because G has been given to glycine, so on and so forth. Say phenylalanine, so as the name sounds phenylalanine, so F has been given to this. So that is some kind of a way of looking at it, but this set of symbols, 1 letter symbols, are accepted and then used globally. So there is no ambiguity on that. So if I want to write a peptide or a protein, I can use these symbols to write the sequence of them. So that we'll be talking about in the later lectures there. Okay, so that's how do we represent amino acids. Now, so what we've done so far is we've just seen what are amino acids, how do we, what is their structure and how do we represent them. I haven't talked a little about their nomenclature. Now let's move on to the classification. How do we classify alpha amino acids? There are 20 amino acids and we know that they have got a very diverse kind of structures and all, and we find that there is no unique way of classifying alpha amino acids. There are a wide variety of them. There are different bases. The first basis is the genetic basis. We'll talk a little while from now. The second is the nature of side chain. The third one happens to be the dietary source. Now let's start with the classification of alpha amino acids on genetic basis. On genetic basis, there basically are 2 classes. The first one happens to be coded or primary protein amino acids. As the name suggests, coded. What it means is, so these are the ones, the amino acids for which there is a code, the genetic code is available there. And secondly, these are the ones which are incorporated one by one in the protein as per the instructions contained in the gene for that particular protein over there. That means I mentioned earlier that a large number of amino acids are there. We talked about 20 amino acids, then there are some other amino acids and there are many more of them. So if I want to just classify them, so there are 20 amino acids which are coded because they are the ones which have got genetic code. So from the DNA they will be translated to the proteins there. So they will be incorporated in the ribosomes and things like that. Now, so they are also called as proteinogenic amino acids. That means they are the ones which will be forming proteins. The second class here happens to be, as obvious, which are non-coded or non-proteinogenic amino acids. So these amino acids are present in the cell but are incapable of participating in the protein synthesis in the ribosomes. So coded amino acids are the only ones which will be becoming part of proteins when they are being formed in the ribosomes there. Now the 20 coded amino acids are the ones which I just mentioned earlier. So the same list holds true here also. Nowadays, recently in past few years, 2 more alpha amino acids by the name selenocysteine and pyrrolysine have been found to be existing in some certain few proteins there, and these also are found to have genetic codes. Basically, they also are in a way the coded amino acids. That is, and the codes happen to be UGA and UAG. But these are the amino acids which do not get incorporated into the proteins in the usual way as those 20 coded amino acids come into being. So they are the new discoveries which has been found recently. Cysteine basically is found in number of enzymes, for example, glutathione peroxidase, whereas pyrrolysine is found to be present in certain microbes that produce methane. This is just for the sake of information, that is, though we talk about 20 amino acids, but we know that there are 2 more added to the list, but there more information is coming on these amino acids. Okay. Now, let us move to the next basis of classification, that is the nature of side chain. So, on the basis of nature of side chain, there are 3 different classes, or you can say 3 groups are there. The first happens to be non-polar or apolar amino acids. Second happens to be neutral or uncharged polar amino acids, and the third one happens to be charged polar amino acids. Let us look at each one of them. Now, non-polar or apolar alpha amino acids, very obvious. That means the side chain happens to be non-polar. That means primarily there will be aliphatic side chains there. So, look at glycine is one exception, because in the side chain we have got only hydrogen atom, because glycine is exception in more than one ways. We will talk about it as we move further. Right here, so glycine is one amino acid for which the side chain happens to be just a hydrogen atom, so it is non-polar. Alanine has got a CH3 group, say valine has got isopropyl, and so on and so forth. So, glycine, alanine, valine, leucine, isoleucine and proline, so all of these belong to because there are aliphatic side chains are there. Proline also belongs to this group here, because it does not have polarity in the side chain there. Proline also has got one more kind of uniqueness to itself, because this is only one amino acid which is not an amino acid in true sense of it, because in this case what happens is, if you look at, generally we got NH2 here, this is my carbon atom here, this is CH alpha, this is my COH, and this is my side chain here, so this is alanine here. So, this is my common thread in all amino acids there, that means NH2, CH alpha and then COH group. So, in this case what happens is, in case of proline, the amino group here, this it gets into, that means there is linked with the side chain here, that means, because otherwise this amino CH and COH has to be there, but this amino group is a part of the side chain as well. So, now this actually becomes a imino group. So, if you really ask me, one should call proline as a amino acid rather than alpha amino acid, but it is collectively used in the same way as other amino acids there. There are 4 more amino acids which belong to this class. They happen to be methionine, cysteine, phenylalanine and tryptophan. Here again, so this is SH group here, but it still is non-polar. So, that the prime thing here is that they are non-polar, because it has a very significant implication in terms of structure, because these side chains which are non-polar or aliphatic in nature, they will be hydrophobic. So, when the protein structure comes into picture, how does the protein fold, what kind of shape does it require. So, these interactions will become very pertinent over there. So, these amino acids have a role to play, all amino acids have a role to play in terms of their determination of the structure of proteins. We will talk more about it as we move further. The second class is neutral or uncharged polar. That means, as the name suggests, the side chain is neutral, there is no charge separation, but still there is small amount of polarity in that. So, there is some kind of a polarity coming into picture. For example, say tyrosine, it has got, so this is my CH2, a benzene ring here, and then this is OH, it is basically phenol part of it here. So, this is phenolic group OH. Threonine, this is my OH again, serine also there is OH, and so on and so forth. So, we find that these amino acids, the side chains are not charged, but they have got some kind of a small polarity in that because of the functionalities available in those side chains there. The third group is charged polar. That means, now the side chain has a charge, and of course, if it has a charge, it will be polar. The example happens to be histidine, arginine, lysine, aspartic acid, glutamic acid, the 5 of them. Lysine is the one, this is my typical backbone here, the side chain there, CH2, 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 the 4 of them, and then there is a NH2 there. So, this is the one which is causing it to be, because NH2 is going to pick up a proton, become NH3 plus, so it will be charged there, it will be polar. So, this is basically is an example of a basic amino acid. Even glutamic acid you see in the side chain we got a CH2, we got a COOH. There is an additional, because besides this NH2, CH, COH, we got a COH group in the side chain also. This is again is a carboxylic acid group there. So, this will be again polar. Okay. So, this is the one way of looking at the amino acid. Probably one classification could be acidic and basic amino acids. So, these two are examples of acidic amino acids. Lysine and ornithine, they happen to be examples of basic amino acids. Even histidine is a basic amino acid. So, is arginine, because the side chain happens to be basic in nature. Okay. So, that is the way we classify amino acids. Okay. Now, having done talked about the classification, let us now move on to yet another way of classifying, that is on the basis of the dietary sources. Let us look at what do we mean by this. We know that amino acids are synthesized in the body. All living organisms, plants, animals, they all synthesize amino acids. But there are certain higher animals like human beings. So, they are not able to synthesize all the amino acids, because our body needs all the amino acids for a variety of functions, because we need a lot of enzymes, we need a lot of proteins, so many of them are required. But our body can synthesize some of the amino acids required for that, but others are not being made in the body. So, we need to have some dietary source for that. And on that basis, we can classify amino acids into 3 groups. They happen to be essential alpha amino acids, semi-essential alpha amino acids, and non-essential alpha amino acids. Let us look at each one of them. The essential amino acids, as the name suggests, so these cannot be synthesized by the body, they must be provided by the dietary source. That means they are essential to be present in the dietary sources. The food we take must have these amino acids. If they are not there, if we are taking food which is devoid of these alpha amino acids, we will be having a lot of implications in terms of health and all. Now, the essential alpha amino acids, they differ from species to species. For human beings, there will be certain group. For other animals, there may be a different set of amino acids which will be essential there. So, their food will need those amino acids. For human beings, there are 8 alpha amino acids which belong to this category. They happen to be leucine, isoleucine, valine, lysine, methionine, phenylalanine, threonine, and tryptophan. So, there are 8 of them. You must have heard about lysine rich bread. Now, a lot of breads come which are rich in lysine there, because that has been added from outside, so that when we consume that bread, you are taking lysine which is required by the body to prepare proteins in it. Now, the second class is semi-essential amino acids. Semi-essential, what does it mean? What does it mean is the following. These amino acids, they are the ones which can be synthesized in the body, but they are synthesized at a rate which is not sufficient enough. That means we need certain amount of amino acids, certain rate is required, they are not formed at that rate. So, it is advisable that these are available in the diet, not necessarily, they are not essential to be present in the diet, but if they are there, they will definitely contribute to the well-being of the organism. And the 2 amino acids which belong to this category are arginine and histidine. Some of the authors even club these 2 amino acids along with the essential amino acids, and then they maintain there are 10 amino acids which are essential. But you must distinguish between the 8 which I mentioned earlier and these 2 which are being synthesized in the body, but at a rate lower than what is required. Now, so they need not necessarily be present in the diet, but if they are there, they definitely are advisable or desirable. The third category is, okay, before I move to the third category, let me make a mention of 2 very important amino acids, cysteine and tyrosine. They are very ambiguous in terms of the classification as we are talking about, because these can be synthesized in the body, there is no problem on that, but they consume, that means cysteine to be made, that uses methionine, but tyrosine uses phenylalanine That means we need phenylalanine to prepare tyrosine. And these two amino acids, methionine and phenylalanine, happen to be essential amino acids. So, the body can make cysteine and tyrosine, but it uses two important essential amino acids for the purpose. So, it is better that if these are present in the food, as this will deplete otherwise the two essential amino acids. Suppose they are present in the diet, it is always good. Otherwise, what will happen is they will consume these two amino acids which are essential, they are not being made in the body, that means we will have to provide from outside. So, if they are there, that is really great.
Okay, this is all about that is amino acids and related facts. So tomorrow it is the last class for the biology and in chemistry. That is we'll read and the next unity of that on. Thank you very much. Good night.